Hello everyone. Hello everyone and welcome to this audio podcast. As part of the prompt series. This audio podcast is part of the Brawl in Rumble, and this audio podcast as part of the atomic headline, and this audio podcast as part of a Storm Robotics production. So in following an audio podcast looking at NASBA as a form of investigation, as a form of particular molecular investigation for and around microorganisms, or detection detection of microorganisms. Now looking at investigation looking at investigation in around a molecular methodology investigation, referred to as strand displacement amplification, or SDA. So SDA is based on the ability of polymerases to initiate DNA synthesis at a single-stranded nick within a target molecule and displace the nick strand during DNA synthesis. The displaced DNA strand serves as substrates for additional nicking and displacement. The methodology has been incorporated into for example, varying assays for the microorganism Trichomatis and inner gonorrhea. There are commercial kits or commercial kits or that utilize in tube detection of the amplified products. Um, at the same time, there is also chain reaction, a ligase chain reaction, or LCR. LCR utilizes four oligonucleotides and results in exponential accumulation of reaction product. In standard LCR, oligonucleotides hybridize adjacent to each other on on each strand of denatured DNA target. A thermostable DNA ligase seals the nick between the oligonucleotides. Each ligated product, along with the original target, can serve as a template in subsequent rounds of denaturation, annealing, and ligation. LCR has been applied to the detection of microorganisms, and again, in particularly around C. trichomatis, Additionally, nucleic acid amplification techniques or investigations in diagnostic microbiology. Some of the sort of disadvantages, if you will, and this is in around the use of both in around both the use of NASBA and LCR, and this can include the detection of clinically irrelevant levels of ubiquitous microorganisms. That is to say, other microorganisms can form part of the diagnostic. Sorry, part of the diagnostic analysis, and as part of the diagnostic analysis, in addition to the main sort of the main microorganisms that are to be detected. There can be contamination of samples with amplified products, and there can be cross contamination of clinical samples, and there can be inhibitors of the application of the amplification reaction. Some of the solutions, however, some of the advantages is that in around nucleic acid amplification investigations in diagnostic microbiology, can include the selection of an appropriate clinical samples for analysis. There can be quantification of amplified products, and there can be separate pre and post amplification areas. There can be use of enzymes such as uracil and glycosylase or UNG, and appropriate, and there can be appropriate positive and negative controls, and use of precautions such as filter tips and dilution of positive control in negative sample, or use of internal control to estimate of internal control to estimate inhibition. So that draws this audio podcast to a close. This audio podcast as part of the prompt series. This audio podcast is part of a ball and run ball. And this audio podcast as part of an atomic headline. And this audio podcast as part of a Storm Robotics production.